The Weinreich lab at Brown University uses antibiotic resistance genes to study how a gene can evolve in bacteria to give rise to increased fitness. Today we are going to learn about the exciting world of gene evolution and discuss science philosophy with our guest, Dr. Weinreich himself. Hello and welcome to RadioBio. My name is Craig Ennis.
I'm Thaddeus Sayre.
And today we're joined with Dr. Daniel Weinreich. He is an associate professor at Brown University in the Department of Ecology and Evolutionary Biology. Thank you for joining us today.
Thanks for having me.
So to start off, who are you and what do you study?
Well, I am a professor of biology. I'm interested in evolution and natural selection. I'm particularly interested in the kind of simplest mathematical models of how evolution works, how natural selection and random processes and mutation interact to change the genetic composition of a population over very short periods of time. We take that as a proxy for what's gone on in populations on Earth since the beginning of life. I don't know, 3-point-something billion years ago. But we don't make any attempt or claims to study super long-term processes. So we work on a small number of mutations over the time scales that we can realize in a laboratory population.
So do you work with like a model organism? Is there a specific one that you...
I would describe myself as a theoretician.
Okay.
We work a lot, but we do have a lab and we work a lot with microbes, with bacteria, E. coli. We've done a lot of work with E. coli. We have 2 big projects right now in the brewer's yeast. We've also worked with a kind of virus that is a threat to bacteria, though not to humans. But what these organisms all have in common are some technical facilities, some technical opportunities that let us construct experiments that are in alignment with our theoretical framework. So we write down some algebra, necessarily making a ton of simplifying assumptions. And then we want to ask, okay, we assume certain values for parameters in our theory. What actually goes on in biology? So these organisms we can grow in population sizes from 10 individuals to 1 billion easily. We can manipulate their DNA and introduce mutations that might be interesting. We can sequence their genomes very easily. We can control the environment in which they live, which is a really important element for us because natural populations like fruit flies or mice or humans live in very complicated environments that impinge on their evolution in ways that are essentially unknowable. And lastly, we can freeze these organisms and then thaw them out and compare evolved strains to their ancestors and see in what way they've changed.
It's very exciting.
That's a lot of cool tools. When you say we, can you just clarify who you? Is
this
an institute or?
I run a small research lab at Brown University. I have a small amount of money from the federal government to employ a couple grad students and a couple postdocs, and lots of undergrads. But really, when I say we, I mean the intellectual community that I operate in. So I have colleagues around the world, not like a dense number of them, but I have a few friends around the world who are interested in similar questions, and we all kind of take advantage of these same opportunities. It's really a great time to be in this field because the theory is really developing rapidly, and the experiments and the technology with genome sequencing and related kind of technological breakthroughs in the last 20 years, it's really a boom time for the study of evolutionary genetics. It really is.
So I see you've done a lot of work studying the Beta-Lactamase gene. Can you tell us what that is? How does it work? And what does that mean for everyone?
So Beta-Lactamase is an enzyme, and its job is to digest drugs, antibiotics. So drugs that it digests are all called beta-lactams. That's why this enzyme has a name that sounds so similar. It digests beta-lactams. Penicillin is an example of a beta-lactam that everyone's heard of. So is amoxicillin, which my kids got when they were young. And there are more recently developed drugs that are in the same chemical family. These enzymes render the drug biologically inert. So these drugs kill bacteria. They interfere with biological processes the bacteria need to survive. And if you expose bacteria to the drug, they die. But if the bacteria has the gene for this enzyme, the beta-lactamase, then they can protect themselves because the beta-lactamase that's just, floating through their world, when it comes into contact with the enzyme, it's digested and it's rendered inert.
Okay, so how have you used the beta-lactamase gene to study evolution?
Yeah, actually, since we're at Merced, let me give a shout out to my colleague Miriam Barlow, who's here and whom I met a few years ago, more than 10, who turned me on to the system. I had this, I told you guys earlier that I'm interested in kind of linking theory with experiments. So I had these theoretically framed questions about what we call evolutionary or mutational trajectories. That's to say, sequences of mutations that over time allow an organism to get better at doing whatever it does. So I had some very simple questions in mind to which there were not good empirical answers.
Okay.
And in particular, what I was interested in is the following. Suppose you know that some combination of, say, 5 mutations together give the organism some big boost. And just to cut to the chase, in the case of beta-lactamase, there are 5 mutations that allow bacteria to survive in 100,000-fold higher concentration of a particular beta-lactam antibiotic relative to the concentration that kills them without those 5 mutations. So everybody's got the gene, but the gene is evolving, and these 5 mutations together allow the bacteria to survive at 100,000-fold higher concentration of the drug. So that's a huge signal. And my question was, how well do those 5 mutations play together? So at one extreme, you could imagine that I have 5 mutations. I put in any one mutation and I get 20% of the boost, 1/5. And I put in the second mutation and I get a second boost of 1/5.
A purely additive model.
A purely additive model. It doesn't even matter which mutation. They're all interchangeable. So that would be, that's sort of at one limit. And in that kind of a world, if I can put it this way, life for natural selection is very easy. There aren't a lot of traps. There isn't an opportunity to evolve to, say, a dead end. At the other extreme, you could imagine a world where you don't get any payoff until you have all 5 mutations together. And if that were the case, it would be very difficult for natural selection to evolve these 5 mutations, to discover and keep these 5 mutations, because no subset of them does anything. So natural selection can't tell what it's looking at until all 5 come together, and the probability of any particular specific set of 5 mutations appearing all at once is really, really low.
Right.
It's analogous to, I think of it as being analogous to the difference between building a stone wall and a stone arch. So if you want to build a stone wall, you add one more stone, you've got one more unit of wall. It doesn't matter which stone you pick up, you just keep adding them, the wall gets higher. That's good. You want to build a stone arch, you put up some kind of wooden scaffolding, you pile up your stones. You don't get a stone arch until the keystone goes in, and 4/5 of a stone arch is no stone arch at all. And we did not know in any system. Yeah. Oh, good. I think it works. We did not know in any system what the challenges were for natural selection. No one had ever thought to ask that question. And I met Miriam and I said, I have this question. And she was finishing her Ph.D. at the University of Rochester, and her professor, Barry Hall, had been working in beta-lactamase. He'd been asking related questions, but hadn't really gone this far with it. And she said, you should look at beta-lactamase. And that's what led to that 2006 paper. Awesome. And the punchline is the truth in that system is somewhere between those two extremes. So you can't just throw those mutations together in any order you want, but in some orders you can get in some orders the mutations will each one improve drug resistance. And the point is that's the property that natural selection can see. So if your mutations are not improving drug resistance or fertility or ability to find food or whatever, then natural selection doesn't know what good they are. And in this system, there's enough of a signal that natural selection can make progress. But we didn't know what the answer to that question was.
So did you limit your analysis to only these 5 residues that are known to be like have fitness effects, or did you look across the whole gene?
Yeah, so it's a really good question. We only looked at those 5. And in every case, the effect you get from adding a mutation depends on what the background is. What other mutations are already there? So I think I said one way of thinking about the study was to ask how well do these mutations play together? And the answer is how well they play depends completely on who they're being asked to play with.
So the total genome, the complement, so the rest of the genome, what you're calling is the genetic background, that has a set of genes that may or may not work together well. And every particular mutation in a beta-lactamase gene has to work well with all these other genes.
No, actually, we held everything else in the genome constant. So when I say background, I mean the other 4 mutations. So I'm going to put in, so one of them changes glycine to G238A, alanine, G238S, serine. Glycine becomes serine at position 238. And that improves drug resistance. But the amount by which it improves drug resistance depends on the presence or absence of the other 4 mutations in the gene.
And that's an example of epistasis.
And that's an example of epistasis, right? So thank you. When I said the effect that a mutation has on whatever the trait is of interest, if that effect depends on genetic background, that is epistasis. That's the definition of epistasis.
Cool. So you mentioned earlier evolutionary trajectory.
Yeah.
So could you explain to our listeners what that is?
Yeah.
And how does that play into this research?
Yeah. So we have 5 mutations. And you could imagine that you can get from the starting point to the ending point by adding, let's just number them left to right, 1 to 5. So you could add mutation 1, and then mutation 2, and then mutation 3, and then mutation 4, and then mutation 5. That would be one trajectory that would carry you from the beginning to the end. But you could also imagine adding mutation 2, and then mutation 3, and then mutation 4, and then mutation 5, and then mutation 1. So the end point is the same, but the sequence of events differs. And so that's what we describe as a trajectory, is some particular sequence of events.
Okay, so are there any limits to the sequence of events that could occur?
If you have 5 mutations, then in principle, you can have there are 120 trajectories, 120 orders in which you could add those mutations. The way to think about that is starting from the beginning, you've got 5 options. Once you pick one of those, you've got 4 remaining options. Once you've picked that one, you've got 3 remaining options. Once you pick that one, you've got 2 remaining options. There's only 1, so that's 5 times 4 times 3 times 2 times 1. That's 120.
Okay.
So there are 120 trajectories if you just think about the mutational process. And we did not have any data in any system that told us what selection thought. And the punchline of the paper was, of those 120, only 18 of the orderings have the property that drug resistance goes up at every step. And that's, as I said a few minutes ago, that's the only thing natural selection can see. Natural selection is intrinsically profoundly short-sighted. It's like Wall Street. What have you done for me lately? So if this mutation doesn't, to use the metaphor, pay dividends now, natural selection will take a pass.
Okay.
So how did you decide whether or not a sequence of amino acid substitutions is good enough for natural selection or not? Do you look at, it was just a selection experiment? You had microbes growing in a certain concentration and you upped the concentration because you added a mutation and you tried to see if they were still surviving?
No, that's what people had done. That's how we identified these 5 mutations. And now when I say we, I mean the community. I did not do that work. But another group, actually Barry, Miriam's Ph.D. advisor, did a key experiment. But people had identified these 5, and he contributed to this, these 5 mutations as jointly giving this big effect, this 100,000-fold increase in resistance, but no one had asked about the orderings. So I haven't had a chance to tell you what my training is, but my undergraduate degree is in computer science. And I used to give this talk. You too?
No. I wish.
When I did the work and I was on the road giving this talk, I always said, without any kind of irony, I did what any good computer scientist would do. There are 5 mutations. That's only 2 to the 5th combinations, 32. I just built them all. I told you that one of the reasons we like microbes is that there are these technical opportunities, and one of them is genetic engineering.
Yes.
Even easier nowadays, there's a lot of excitement in the community. You may already have interviewed somebody who uses CRISPR technology, which allows you to engineer in specific mutations into eukaryotic genomes, which are profoundly more complicated than bacteria.
Yes.
But for many years, there's been technology that allows you to put any mutation you want into a bacterial genome. And so this is technically very simple work. I had an undergraduate who spent the summer building these. I mean, I built one or two to make sure I knew how to do it, and then I supervised him, but he did all the work. It's just PCRs and sequencing. I have a student in the lab right now who's building some of these, an undergraduate. The technology is really accessible.
So you have this library of mutations, and then what?
Well, it wasn't even a library. We had pure culture. So you engineer in the first mutation, this G238S that I mentioned earlier. So in the freezer, I have 32 tubes, one for each of these variants. I just, in pure culture, measure how well it grows in the presence of the bacteria, in the presence of the antibiotic, another really cut and dry technology that Miriam turned me on to. I mean, this is stuff that medical microbiologists are very concerned with. A patient presents to the doctor, they presents at the hospital or to a doctor with an infection that's not responding to an antibiotic. So they'll culture the bacteria and measure its resistance. There's a well-established protocol for doing that, and we just adopted that protocol.
Are we talking about like MIC?
We're talking exactly about MICs, yeah.
That means minimum inhibitory concentration.
That's right. So it's the lowest concentration that blocks bacterial growth. And the way you do it for any one of these 32 constructs is you make a series of media that have a range, each of which has a different concentration of the antibiotic, and you inoculate them with your strain, and you simply ask, what's the lowest concentration of antibiotic at which the bacteria fails to grow? It's done visually. It could not be. Undergraduates do that routinely in the lab too.
So what kinds of questions are still unanswered in your system of study?
Beta-Lactamase is called Beta-Lactamase because it hydrolyzes beta-lactams.
Yes.
And it was purified using technologies that you start with a total cell, you grind it up, you get a bunch of proteins or enzymes, and you find the fraction that has the beta-lactamase activity, and then there are biochemical techniques to purify the fraction, and you just follow the fraction that has the beta-lactamase activity until you have a fairly pure sample. And then, in 1964, you do Edmond degradation or some protein sequencing technology to figure out what you got in your hands. But that doesn't mean that beta-lactamase is the only thing, that hydrolyzing beta-lactams is the only thing that a beta-lactamase can do. And in my view, that's generically true for all enzymes, that they have a name because that was the activity that was followed by the biochemist who purified the thing in the first place and then cloned the gene, blah, 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 blah. But from the cell's point of view, that's just a label. And it's reasonably well established, I think, that many enzymes will bind a different substrate and maybe do the same chemistry, but to a different substrate at a rate that's 1/1,000,000th the rate that the biochemist used to purify the thing. But from the cell's point of view, maybe that's important. So that's called enzyme promiscuity. And I have an idea that I don't have the technology running in the lab, so I can't even imagine writing a grant to do it. But I have an idea for how to get after the question, what does natural selection think about these promiscuous activities? Like, you could imagine that natural selection doesn't care. So, and the way that would work is you've got a bunch of variants of beta-lactamase out in nature, and you would ask, well, you've got a single gene that can fold into multiple conformations, and each of those conformations can do something a little bit different. Now, what's natural selection going to do with that? Well, now I do need, right? Natural selection primarily operates on genetic variability. So let's imagine I've got a second allele, which has a slightly different conformational ensemble. It mostly folds into the same states as the first variant, and thus mostly has the same kind of fingerprint of enzyme reactions it can catalyze, but it's slightly different. Natural selection might have an opinion. So I've got two competing variants, and the only difference between them, they both are great beta-lactamases. They both have the same MIC. Okay, but one of them has a 1/1,000,000th fold activity in some other on some other substrate. It hydrolyzes something else at 1/1,000,000th of the rate that it hydrolyzes the beta-lactam. And the other one has that same second activity, but 5 times stronger, right? Now, there are these two things. So let's imagine these two things competing in the population. They're equivalent with respect to their named activity. They protect the bacteria just as well against the drug, but one of them has a slightly higher activity, albeit extremely low. Well, natural selection might not like that. That might be a meddlesome activity, in natural selection, the organism says, I didn't want that other thing hydrolyzed. I'm not able to grow as effectively, so natural selection would suppress the variant that has this slightly higher secondary activity. Natural selection might say, gee, that's awfully handy. Like that actually is something that the organism can take advantage of and enrich for the variant that has this. These are really, in my imagination, these are really low-level activities. But my intuition is natural selection might have opinions on the value of those additional activities. My intuition is those additional activities are just part of the process of being an organism. It's not like an engineered thing where there's a pipeline where we do this here and we do that there. Mm-hmm. It's just a bag of mush.
Right.
And so I have some ideas for how to get after that experimentally. But
so imagine this is your last chance to teach the world a single concept in biology. What would you say to them?
Yeah, that's the one question on your list.
That was the tough one?
That I was like, whoa. And I was talking to my girlfriend about it this morning. She said, that's pretty philosophical. And honestly, that's where I go with it. Because my intellectual passion for evolution is motivated by a strong sense of aesthetics. Like I don't pretend that there's a purely scientific motivation, and I just am. Well, let me put this into a little more rigorous context. I told you I was an undergraduate computer science major. Mm-hmm. And as an undergraduate, I had the great fortune of walking into a graduate seminar at my undergraduate institution taught by a guy who had invented something called genetic algorithms. Okay. And so these are computer programs that evolve. Mm-hmm. And the original application was to use them to find computer programs to perform tasks that are so complex that we don't really understand how to do them. Mm-hmm. So if you want the next digit of pi, I can write down an algorithm that will just spit them out one after the other. But there are lots of challenges in the world, like, playing games like chess and checkers, mm-hmm. where at least when I was an undergraduate, which is the early 80s, they hadn't been solved in a mathematical sense. So there were only heuristics. And problems like that are really hard to program. And so the idea here was, let's get a population of computer programs that have each of which has a slightly different set of heuristics to solve the problem, and just play them against each other and find the set of heuristics that's most successful. And there would be, so there's a population, just like in my field in population genetics, and there's variation, and there might be some kind of variational operator that introduces new variation, maybe by mixing the left half of one heuristic with the right half of another, which is inspired by genetic recombination.
So you're talking about your genetic algorithm is also a selection.
Yes, okay. That's right. So you have to have some kind of they call it an objective function that allows you to tell which of all of the members in these populations are doing better and which are doing not as well.
So like a fitness.
Like analogous to a fitness function. That's right. And I was a computer scientist. I was taking classes in data structures and algorithmics and stuff. And this possibility that this is essentially, this is Darwin's paradigm, but I saw it as an algorithm. Like there's mutation and recombination and reproduction and survival, and mutation and recombination and reproduction and survival. It's just a loop. And so I got very interested in what that algorithm could do. And that's still what I'm really interested in. And so that's kind of like an intellectual motivation. But the thing that really makes me so excited is to couple that kind of intellectual traction with the extraordinary diversity and complexity of life on Earth. So somehow in my last lesson to humanity, that would be a message I would try to communicate. But mm-hmm. that's why I was saying to you, my answer is kind of philosophical, and I don't really know how to put that into a 50-minute lecture. Yeah. Or a 50-minute podcast. But that, mm-hmm. like that to me, that there can be a material explanation for both the world that we experience and the fact of our experiencing it, mm-hmm. blows my mind. So that would be what I would want to communicate.
Very cool.
Very cool.
Is that like the idea that if you knew all the algorithms for everything, right, you could calculate the perfect statistic, like that one event would happen. But you're talking about but how do we explain what we observe now, right? It's because if we could do it, we could use these processes, simulations maybe, and we could find out maybe how it happened, at least theories.
Yeah. I mean, I'm not that,
No.
the piece that I'm resisting is the idea that you could have perfect knowledge. Like these processes are so dependent on stochastic components, randomness, like which mutation happens next, who knows? Yet my beta-lactamase experiment tells you that if this mutation happens, then the next 4 mutations, like there are forks in the road, and if you just by chance take the left fork, the next period of evolution will be different than it would have been if you'd taken the right fork in the road. That's not actually that clear in my data, but other data sets that are like it, even in beta-lactamase, totally make that point. There's also a lot of randomness in populations with respect to who actually successfully reproduces. So evolution is a process that occurs not to individuals, but to whole populations, right? I'm born with the genes I have, and for better or for worse, these are my cards and I'm going to play them out. But the population over time changes in its genetic composition. And a part of that, a key part of that is who gets to reproduce. That's where the next generation comes from. And you can have the greatest allele in the world, let's say for resistance to antibiotics, but you fell into an open manhole and died. And that good gene didn't save you. So there's a lot of randomness at a lot of levels. So the best we can hope for, I think, are sort of statistical averages and things of that nature. So that was the part to your question that I was wrestling with. Was I don't want to imagine that we can have certain knowledge, but I think we can learn a lot. Yeah, I think we can learn a lot. So, I mean, relatedly, I have to admit that I'm kind of more interested in framing good questions and less interested in answering them. There are a lot of things that are interesting, that are curious about the world that I don't know how to begin to answer. It would be great if we could sprout wings. We could fly around, and that would be pretty awesome. And yet that hasn't happened and doesn't seem like it's going to happen. But why? I don't know. I mean, people have theories, but that's not a place where I feel like there are experiments that one could do. So a large part of what gives me joy is identifying places where there's theory that can guide experiment. Almost that's more fun than what the answer actually turns out to be. So that there are 18 paths instead of 38 or 120, like whatever answer there was would have been neat because the question was well formed and the answer will be compelling no matter what.
Do you have any recommendations for how to start asking the good questions? So you recommended looking to the literature, but do you have any other advice for how to formulate a good question?
No. I mean, that is really, the $64,000 question.
Yeah.
Speaking of answering a question with a question, right?
Yeah, as someone that's new to a graduate program, learning to ask the good questions is something that I've been trying to work on. So I guess more literature.
Yeah, the literature is important. I think another good idea is to talk to people, and most importantly, and this is possibly the most important lesson I learned in graduate school, to ask plenty of questions of other people. And this also connects with what I was saying earlier about how some of the students I've enjoyed working with the most really have a confidence that's ego-free. So they're perfectly happy to say, I don't understand. And the best collaborations that I've experienced are with people that have a shared interest in a problem, and everybody just kind of drops their guard and starts chiming in with ideas. So that's not really a thing you can bottle, a technique you can deploy on demand or on command. But I would say, my students come back to me with questions and we sort of brainstorm together, and that's the way it works with my collaborators as well. And so that's, I don't know, this idea that drove the 2006 study, I don't exactly know where that came from. That was mine, but I don't know what I was thinking about the minute before I was thinking about that. But it's now it's a cottage industry. Like there are lots of people doing those studies. And so we're getting these lovely data sets that like mine. So maybe in another system, there were 6 mutations. And so if there are 6 mutations, then there are 6 times 5 times 4 times 3 times 2 times 1, which is 720 possible paths.
Yeah.
And maybe it's a different fraction of paths that are accessible. And we can kind of take averages and look for kind of trends and patterns across these data sets and try to learn kind of deeper things about biology. Like, for example, the data aren't very strong in this dimension yet, but the little bit that I've seen suggests that these patterns of epistasis, the constraining consequences of epistasis, are much more common among mutations within a single gene, and less so in mutations between genes. So that sort of reinforces an idea that's old, which is that genes code for proteins, and proteins kind of are these atomic objects that do their thing. And it's like a subroutine in computer science where there's a well-defined interface, and what goes on inside of it, you don't have to know. And so if you change one component in a big software engineering package, it shouldn't break other things. But if you change one line, it might break that one module.
Because that module's inputs and outputs, that function that it does, it remains invariant.
Right. So
it doesn't really matter inside how it's doing.
Let me just point out that I'm bringing a lot of suppositions about the way biology ought to work, which aren't that well established yet, in my view. So, as I say, there aren't great data sets that compare the kind of epistatic patterns within a gene among mutations within a gene to epistatic patterns among mutations between genes. But the data sets we have are consistent with that idea of modules. I think there's a great temptation to look at molecular biology the way an engineer would, and that's good. Like, those are good tools, but it's not always sufficient to understand what's going on, because evolution is not an engineering process.
So if our listeners want to get in contact with you, what's the best way?
Email. Yeah, so it's just my last name, Weinreich @brown.edu. But yeah, I'd be very happy to talk to anybody about any of this stuff. I mean, this is the curse of being a professor, is you can talk a lot longer than anybody wants to listen about your favorite subject. That just comes with the territory.
Mm-hmm. All right. Well, thank you so much for joining us today.
Sure. My pleasure.
Super.
RadioBio is supported by the Quantitative and Systems Biology Graduate Group at the University of California, Merced.